announcement. Two things. First is that you have to vote for the date of the final home assignment. Those of you who still haven't done it. I will close it soon, so please hurry. And the second thing is that the third and last home assignment about Huntington's disease will be published next week. Due to the very few requests to extend the deadline, so we cannot do it because it's not fair towards other students who submitted on time. So please try to do it on time. Don't wait for the last minute, okay, and then something happens and you get stuck. Okay, so it will be the last home assignment, and I think that afterwards I'll close the poll. Okay, we'll see what's the date for the last
Okay, so good afternoon. First, I want to ask you, don't check your phone when I'm teaching. I know it's annoying for me to say. If you want to check your phone, you go out and then you come back. I have no problem. But please don't do it because I feel, okay? I feel it's a lack of respect. I know it's a new generation thing, so that's why I say it and not complain later. Okay, guys, so today I'm going to, I don't know if you remember, I gave a class a few months ago or something like this, and now I came back to give you some of the basic background after many of the classes by guest lecturers. So I'm going to basically talk about two topics. The first one we'll talk generally about the damage pathways in a neurodegeneration, in neurodegenerative diseases. We don't have a pointer here, no? But I think I brought one. And the second part of the class, or towards the end of the class, I will talk about animal models of neurodegeneration. And again, the main idea here is that you guys ask me the questions, and it's not so much about the contents, but about to engage you on this, okay? So some of the things I'm going to talk more, I mentioned before, but what I'm trying to do is we are going to go through one disease and all the genetic variants that you can have. We're going to try is Alzheimer's, but it's just an example of how you can go over this. Okay, so So how we determine cause effect in neurodegeneration? So as you know, when you take a brain of a person who died from one of those diseases and a normal person, you have many, many things that are different. And of course, it's tempting to say that all these are the cause, but it's difficult to say what comes first, okay? Because we are not, well, luckily, the people die many years after being diagnosed in most cases, so we cannot see what happened just before starting to have this defect or just before having the disease. So it's difficult to say what comes first, what comes second, what comes third. We only can see the end point. And this end point, usually, there's a few common features in all these diseases. Can be Parkinson or Alzheimer's, which has to do with high oxidative stress, with these big aggregates we see in the cells, with the dysfunction of the mitochondria, with microtubule assembly problems, apoptosis, cell death, etc. So you really have a lot of phenotypes or a lot of defects on those individuals, and it's difficult to say what comes first, what comes second. So what is the cause and what is the consequence of neurodegenerative processes? And of course, a typical approach will be to have access to very early cases. And I mean, these will be people who just were diagnosed with Alzheimer's or Parkinson. And so of course, someone can tell me this is already too late, okay? Because if you have been diagnosed, you may already have neuronal loss, and many of these consequence effects may be already there. But even if we can say, okay, but it's better than having a person which is fully developed the disease, it's almost impossible to get access to this type of sample. And this is a lack. Okay? This is because these people are not dead. Because the only way to get a sample if you take the brain and you look at it. But in most cases, the people don't die so early, which is a good thing. So, but we don't have access to this type of cases. All right, keep going. Other thing that you can do is that if you have, let's say, 20 things that are affected in a person with Alzheimer's, you can say, okay, we can take now a mice, and we can perturb one by one of these 20 things, okay? And then we can see whether when we knock down any of those pathways, we now have a similar mice that forget things or have neurodegeneration, etc., etc. But this is a little dangerous because, of course, I don't, you guys, most of you are in social sciences, so that you know some of logic. This is a fallacy. If you have, if you have something that provokes 20 consequences, and then you just, you cannot go the other way around, You cannot say, if you have 20 pathways in the neurodegenerative brain, and then you knock down one of these pathways and you get the same, you cannot say that this is the reason in the first case. So this is a fallacy. This is very common, okay? This is extremely common, especially in medicine. So when you hear, especially in the past, now it's a little better, the research aspect that the doctors, or the medical doctors learn. But this is very, very common in early research in medicine, maybe 40 years ago or something like this, that they will say that they will do this type of fallacy, okay? But still, this type of approach to affect pathways in a mouse model and to look for defects that resemble neurodegenerative diseases is something which still is a powerful thing we can do. So one thing that is very, very common is to look for inherited forms of the disease. And then, for example, I'm sure that you heard from Mona and from other speakers that in Parkinson or Alzheimer, you have most of the cases are sporadic. This means most of the cases, the person who gets the disease, you cannot make, say, okay, it's not a Mendelian trait. It's not something that comes clearly from inherited forms, okay? Usually, maybe his parents or her parents didn't have the disease, or very few people in the family, but this person will get it. But there is a few cases, a small percentage, depending on the disease, can be 2%, can be 5%, can be 10% of cases in which Parkinson or Alzheimer or other neurodegenerative diseases go through families. And in this case, you have specific mutations that can be unequivocally traced or assigned to the disease. And you can know that this mutation in this protein or in this other protein is really the cause of the disease. And in this case, this is very useful because now you know this gene is causing the disease. So you can try to understand why and how. And for this, you can basically make a mouse or make cells that have the same mutation, and you can try to see what happened and how this happened. And you can also try to find genes which are associated with the disease, even if the diseases are sporadic, because you can think that when you have multigenic diseases, you have a specific, let's say you have one mutation cannot cause the disease, but maybe 20 different mutations together can cause the disease. And maybe you get the disease because you get 10 of these mutations from your mother and 10 from your father, but none of your parents will have the disease. So many sporadic cases of the disease can be consequence of still genetic. The disease is still genetic. It's just more complex genetically. And then you can try to see, if you look 1,000 people who got the disease, you can try to see which type of mutation in common these people has, and you can try to trace how the genes which are important for the disease. So I told you, I think that this you have listened many times, but if you forgot about it, I tell you again. Okay? that they are sporadic versus hereditary forms of the neurodegenerative disease. If you go to Alzheimer's disease, most of the cases are sporadic, but you have a small percentage of the people in which Alzheimer's runs in families and is genetic. So usually what you see in Alzheimer's, you see that young people don't have Alzheimer's, but after 40, people who have inherited form of the disease are going to get it, and the sporadic cases appear around 65 years old. So basically, the cases with random families, the cases which are inherited, show up very early, much earlier than the sporadic cases. The same for Parkinson. Some diseases like Huntington only appear, there are no sporadic cases of Huntington. This always run in the family. And ALS, again, most of the cases are sporadic and appear after the 40 years of age. Okay? Now, what is the difference between the sporadic and the hereditary forms of the disease? How we know it's the same disease? Okay, because how we know it's the same disease? Basically, because we can see the same symptoms, the same behavioral symptoms in the person. When these people die, we can look at their brains and we can see that they really look like each other. The same neurons are being lost, the same type of aggregates, etc., etc. So the question is, what is the difference in one case or the other? The similarities, the disease seems to be the same, but still, in the inherited form, is happening much earlier than the sporadic form. Okay. So in Alzheimer's disease, and I think that again, Mona has talked about this, but my intention is not to focus on Alzheimer's, but try to systematically show you the type of pathways which are affected in this type of diseases. In AD or Alzheimer's, you have this type of fibrils, which are extracellular. They are outside the cells. So if you take a brain of a person with Alzheimer's and you stain it, you're going to see these very big aggregates outside the cells. You also have other type of aggregates which are called NFTs, or neurofibrillary tangles, which are intracellular. So these people have not only the extracellular aggregates, but also the intracellular aggregates. And I know that you guys have heard this from Mona before. No? You heard this from Mona, no? Yes? Yes, okay. That the way these fibrils are being done is because of the release of this monomer of A beta, which is a little longer than the usual and which can aggregate. In the case of the NFTs, the main effector here is the protein called tau, that when it's phosphorylated, gets like a ball, like a garbage can, and they aggregate one to each other, and this accumulates inside the cell. In my first class, a few weeks, even 2 months ago, or something like this, I mentioned that sometimes this aggregate, there is a debate whether they are helping the cell or whether they are harming the cell. Okay? But still, the aggregates are always there. Okay? Okay. So this, what was the canonical hypothesis for many, many years was this amyloid hypothesis, which basically proposed that you have this A beta precursor, which is going to be cleaved by beta and gamma secretase to give this peptide A beta. Of course, depending on the cleavage, this peptide is going to be the toxic form or not the toxic form. And now you have many ways in which you can have the same, okay, you can have a person who has propensity to generate this peptide. But now this is not enough, because you can have many ways that can be protective and many ways that this peptide can be more toxic. So number one, you have mechanism for clearing to the brain, clearing from the brain. So any mutation that enhances the clearance is going to give you advantage, even if you produce a lot of this peptide. But any mutation or any defect in the clearance is going to increase your propensity for the disease. The same with degradation. If you have many of these peptides supposed to be degraded, but if you have impairment or mutations that go against the degradation, you're going to have a higher tendency to have the disease, and the other way around. Also, the A beta is not toxic as a one molecule, but it's toxic as an oligomer when a few of them get together. So whatever will influence the property of the molecules to make oligomers is going to go against and or after. You also have these neuroephemeral tangles. These are the intracellular aggregates. And again, whatever will favor the aggregation will lead to death and whatever. No. So what I'm trying to make here is to show you that indeed AD or Alzheimer, like many other neurodegenerative diseases, are not a yes or no. It's a degree. Maybe every one of us has some of it, but depending on secondary and tertiary mutations, the severity of this will show up very late in life, as late as we are dead already, or earlier in life. So this pathway is going to exist in all of us. Just the degree is a quantitative difference between people, not a qualitative difference. Okay? Clear? Any question? Okay. Okay, so this is just, I just put this hypothesis that there is a change in the metabolism of this A beta. This peptide is making different proportions. Depending on the ratio between the 42, which is a bad one, and the 40, which is a good one or normal one, you're going to have a more toxicity. You have effect on the degradation or clearance, which will, okay. So you have everything I told you is here, okay. So the question is, which type of genes are associated with Alzheimer's disease? And here I just going to explain you how people do those type of studies and what they have found and why. I want you to understand why looking at this picture. Okay. So, of course, this movie or this picture starts here, no? With the production of the A beta peptide. So the first mutation people identified, which were linked to Alzheimer's disease, are mutations in the precursor or in the apparatus that cuts the peptide. So whatever is going to make the precursor more prone to be cut and generate the bad peptide is going to make us better or worse. Worse, okay? What I just feel you guys are in a sleepy mode with the eyes open, okay? I understand it's late, but let's try to have, I would not say fun because it will be a shock. But I think it's an interesting thing. I'm trying to convey you guys this, okay? So the basic picture here then is that if you have, you are generating this toxic peptide from a substrate, this long peptide, with these two enzymes that are a scissor. So if you have mutations in the substrate that favor making the bad peptide, of course, it's bad for you. On the other hand, if they favor the good peptide, you are going to have less chances of Alzheimer's. And the same with the enzymes that cut the peptide, mutations that make this cutting more likely to generate the bad peptide is going to happen, will be bad for you. I mean, and this is, of course, this is like the concept I want to put here is this quantitative. If you need to leave the room, this is the concept I want you to learn about, okay? That you need to accumulate so many mutations, but it's a qualitative against quantitative. So maybe there is something that we will be able to do to tilt the ratio, okay? The early onset Alzheimer is very, very low. And mutations in these key genes are account for around 5% of the Alzheimer. And I should say this is a lot, because this means 5 out of 100 people who get Alzheimer have mutations in one of these enzymes. So if we would have the way to fix this could increase. Number one, of course, this 5% will be sort of safe. But as I told you before, this is a quantitative disease with many loci, but the center is here. If you are going to generate less of the toxic AB42, you can diminish the disease much than the 5%. That's why it's so important to focus on this. Yes?
Is there, can you cut out the mutation, or do we still need that gene? For example, we still need tau, do we still need that gene?
Yeah, it's a good question. So yeah, we do need it. So it's not clear why the APP, what exactly the function of APP is, but if you generate mice without APP, they have some synaptic defects. So you cannot just eliminate it. You need to correct it, which is more difficult now. But I think Mona mentioned that there is ways, there is drugs that can change the cleavage efficiency. So it's not at the genomic level changing, but it's a drug that can change a little the efficiency of this cleavage and generate more of the good peptide. But now there are methods for genome editing. Maybe eventually in 10 years, people could correct this or could introduce a variant of the APP which cuts. What is important here is the ratio. It's not the amount of the bad oligomer, but it's the ratio with the good oligomer. Maybe we can increase the good oligomer, and maybe this can. People are doing this to see how to do it, But it's a good question, yeah. So this is the pathway we are going to go, and we are going to try to focus a little on this. So I just mentioned you that you can have mutations in APP, the substrate, or in preselling 1 and 2, which are in charge of cutting these precursors. So this is one level of regulation that can lead to accumulation of the bad eta B. So I'm not going to. I know that Mona mentioned this. So this is just for you guys to have it there. The mechanism of the cleavage by the two enzymes, how this is cut, etc., etc. Okay. Okay. Yeah, I think that this is is redundant with the class of Mona. Just leave it here. Okay. Okay, so this is just sort of summarizing. When you mutate the substrate or you mutate the enzymes, then you can increase the ratio of the bad peptide, the A beta 42 to A beta 40. And most of these mutations are missense mutation. You know what missense mutation is? This is a mutation that doesn't make that the protein disappears, but not just what makes is that the protein now is making something, change the amino acid. So change the efficiency of the cutting, or change the efficiency of the protein, or change quality of the protein. You know that the protein disappears. You know that the protein is not more there, just is that the protein is different. Okay? Okay. Most of the mutations are in PCS, preselling 1. And the mutations in the APP, in the substrate, are near the place where it's being cleaved. Why this is important? Because all this is pointing out that what is important is not that APP is doing something else. It's about the cutting of APP. Okay. So what happens? What happens if you have more APP? Okay. What happens if you have more APP? So there is a duplication of okay. So you have some cases you have duplication of the locus of APP, and this results in much higher rate of Alzheimer's. This now is a dominant trend. Some of the mutations, some of the familiar mutations are recessive. You need the 2 alleles to have Alzheimer's. This particular duplication of the locus leads many times to the disease to be dominant, which is terrible, because this means basically if you get the allele from the sick person, father or mother, you're going to get it for sure. Okay. Okay. I'm not going to go into this. Let's not go into this detail. Okay. So you also have this from before. Okay. So what you can do to change this, okay? We have now mutations either in the APP, in the precursor, or in these enzymes that led to generation of more of the bad peptide and less of the good peptide. So people have tried to do a pharmacological approach. And I should say that nowadays there is a lot of new technique for gene editing. So probably people will be able very soon to change the genome and to fix some of these mistakes very soon. Or not, probably not in all the brain, but at least probably in many cells if we will need to. So I think that now people will start talking about applying this to humans, although it's very new. But until now, people were trying to get compounds that will lower the production of A-beta monomers, okay? And this can be done by many ways. One way will be just to inhibit the enzymes that cut it. But as I said before, this has not been very successful because usually when you inhibit the enzymes, you don't produce more A42, but also you don't produce A-beta 40. And what is important is the ratio rather than the absolute amount. Also, people saw that the small peptide is not the toxic species. So if there is a way that we can label and destroy the oligomer, which is the toxic species, so then this will work. And the rationality for these people is to use vaccines with the peptide, so the body will generate antibodies against it. Okay. So you also can try to. Okay, it's dead. You can also try to stimulate the degradation of these toxic aggregates, but this is indeed very difficult because this aggregate is like a ball of crap. You know, it's very difficult to degrade. Indeed, the cell cannot degrade it, so it's very difficult to find enzymes that could do it eventually. You can also try to destabilize these toxic species. Okay, and you can try to change the specificity of this gamma secretase, the enzyme which cuts, and that maybe will generate more of the A-beta 40. So really, a lot of the efforts are centered on this now. I mean, in the last, didn't succeed, to be honest, most of these efforts in the last 20 years, unhappily. Okay, so so what other type of pathways are important here? What I told you today, okay. What I told you today is, until now, this is a whole pathway, the whole hypothesis. And people always ask, is the sporadic form really genetic? Or the sporadic form maybe just, because when something is sporadic and you cannot explain it based on simple genetics, you always wonder whether it is environment, what you eat, what you do, you know. So there was a few studies for this a few years ago, and many of these studies are the twin studies in which you take twins which have been separated, and then you can do indeed sometimes many big studies, and basically in Alzheimer's disease, and this comes over and over to the same, genetic factors should look like have most of the influence. So this disease seems to be driven by genetics. It's up to 80% of it, okay? Maybe even more in other studies, but around 80% is genetics. Now, the genetics, this doesn't mean that we know how it happens. The genetics is very complicated. You can only see association between people in the family, and that the possibility of having it, if you have a twin, is very high. I mean, this means that it's genetic, even if the twin was grow up in a completely different environment. So the question is, what are the genes, and how complex is this genetic thing? So why we want to identify the genes? Someone can tell me why we don't have cure to the disease. So why we want to identify the genes? Because what? Then we could check the genes and tell the, you will have Alzheimer's. And then what we do, you know? Yes?
Well, everything's not only genetic, you also have the environment. It's not entirely certain what has more
Yeah. No, I think that it's clear. Most of it is genetic.
It doesn't. It's still, you can. Maybe they can get it, but it's the difference between the person gets it when they're 60 to when they get it when they're 80, and also how fast it progresses and stuff like that.
But if you take two twins, and one you reach in a rich family and one in a poor family, they are going to get the same chance to get Alzheimer's. But if you get two brothers raised in the same house, but with different a little different genetics, not completely different, but a little different, they are not twins, they don't correlate at all. So I'm not trying to contradict you. Thank you. You're bringing a good point. It's mainly genetics. It's mainly genetics. But it doesn't mean we cannot influence it. If we know which genes are important, then number one, we can understand how the disease works. Okay. Number two, we can try to, if we know that this is, I'm just making up something. If we know that the disease involves high blood pressure in the brain, then maybe we can try to lower the blood pressure in the brain. If we can know which genes are important, then we can try to inhibit them or changing them, and we can change them with drugs or with habits, like feeding and stuff like this. We know that the people, of course, who live a healthier life have less chance for many of these diseases. We don't know exactly why, and what part of the healthy aspect is. But again, those diseases, if you do a serious study, are almost completely genetic. Unhappily, there is little things you can do. This applies indeed to many diseases, even to many types of cancers. But again, it doesn't mean we cannot do anything. Indeed, if we understand about them, being genetics gives us the advantage that we can isolate the genes and then understand how they produce. And then we can use intervention with environmental factors to delay or to change this, or go against these genetics. Okay. That if you have certain mutations that really predispose you to Parkinson, there is one thing that you can do. Mona told you about this. Some mutations give you very high chance to have Parkinson's disease. If you smoke, then you could delay it indeed. Okay? Smoking, nicotine is something that can delay Parkinson. Now, the disadvantage of smoking outnumber the advantages. But maybe if you have a mutation which is maybe then it's worth it to take the risk. Okay? She mentioned this, no? Or no?
She did.
The protection, eh?
She said that there wasn't an actual positive correlation between the two, that smoking or drinking coffee does not necessarily mean you do not get the disease, just maybe because you have the ability to get addicted to something. Right.
Yeah, I think that the at least in mouse model, the nicotine seems to be also it's stronger than the caffeine. Okay. So how you find these genes which are affected? Okay. How do you find these genes? So imagine you have a population of people which are affected with the disease, and a population of people who are unaffected with the disease. So now you have, of course, the genome is huge. So you have a lot of mutations. You need to try to identify which mutations are in common between the people who have the disease and between the people which don't have the disease. Okay? So I should mention this for everyone. I will not say this. Okay. So then, how you do this study? How you do this? And this is what we call genetic association study. It's trying to see the association of specific alleles, specific mutations between people. So actually, if I take the genome of you or myself, it's not going to look the same. And this doesn't happen. I mean, if you look at the specific base by base in the genome, it's not going to be the same for everyone. What is going to happen is that some of them will be more variable than others, okay? But the variability between people is very big. And this is what we call, it's difficult to call mutation because mutation has like a negative connotation, no? Yeah, I mean, for non-biologists, let's say, has like a negative connotation. But we know that really mutation doesn't mean anything. Most mutation don't have any negative effect on the survival, on the capability to reproduce, etc., etc. Very, very few mutations are negative, and very, very few mutations are positive. But most mutations are neutral. They have no effect. So then what happened in consequence is that most of our, or many of our bases in our genome is different between people. And of course, you have different, what we call a SNP or a small polymorphism. Some of these bases are more common in some people or other people. And you can indeed follow between relatives, the difference will be smaller. So you can, this is a way to do filiation studies, et cetera, to look in a specific SNP or a specific sequences which are going to be, or a collection of mutations which are going to be very similar between brothers or between parents and sons, etc., etc., etc. Okay. So how you do this type of association studies? For this, you basically use these millions of bases which are not the same between us. So some of those may be not good. So if you accumulate 100 of those which are not good, maybe you get a disease. Okay. If you accumulate 100 of them which are better, maybe you are protected against the disease. Okay? Most complex disease involve many, many hundreds of genes. It's not just one or two. I have a question, yeah. Yeah, we'll go now in the detail. What I'm trying to say that the genome of each one of us is not the same. Okay? It's in certain places it's exactly the same, because if there is a mutation there, we will not be alive. But in many places it's not the same, and has small variations that you can call them mutations, but usually we call them small SNPs. Okay, SNPs usually are not good or bad for you. I mean, some of these SNPs may have a very, very mild negative value, or very, 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 very mild positive value. So when you accumulate many of them, you may get a disease, and maybe that's exactly what I was talking now. You have 10 mutations, or call it a specific SNP, coming from your mother that are not good or that will predispose you for Alzheimer, but it's not enough to get you Alzheimer. Then your parent may have another 10 in other places in the genome that is not enough to give him Alzheimer, but you are maybe unlucky that you get those 10 from each one, and now you may get it. In a complex disease, you have many, many small mutations that make the picture. You can also have mutations that happen to you and they were not in your parents. Happen to you when you are a little cell in the belly of your mother, you know, or when you were a little bigger, in some cells, in all the cells, and these are called de novo mutations. So those mutations are not inherited from your parents, but in this case, the disease is still genetic because it's in your genes. It's not a consequence of the environment. Of course, you can tell me that they might have to do with the environment or what happened in this during the pregnancy, okay? But that doesn't really matter. What I'm trying to say, what you do in this association study is try to see of the million of variations or mutations, if you want to call it, we have in the genome, whether a set of them is associated with a risk to the disease or protection to the disease. So what is SHE-WAS? SHE-WAS the genome-wide association study. And this is, involves to look at these millions of variants, these millions of small mutations, if you want to call it, and to try to relate them to the disease. Okay. For this, you need a lot of people, usually in the order of hundreds to thousands. Okay? And you need also a lot of tests. You need to test all those 1 million sites. Most of them will be not informative because most of them will be just randomly distributed between the people. Also, you need to get people. Okay. And this idea is that this type of studies will try to give you combinations of mutations that are important in common diseases, which are usually multigenic, which involve mutations of many genes.
Excuse me?
Yes.
What's the age group of the subjects?
Yeah, this is a good. that shouldn't really matter, you know. But if you are talking about a neurodegenerative disease, you should do it late enough that the people who are your control individual, they're not sick people. Of course, you cannot do an experiment if the Alzheimer are taking 70 years old and your control is 20. They need to be matched. They need to be matched in age. They need to be matched in gender. They need to be matched in general habits. You know, you need to control for some of those parameters. Okay, you need to know whatever you believe is important is equivalent between the two groups. This is a good question. This is a very important control. Now, for neurodegenerative disease, you need to always use all individuals because you know you could maybe find young people that don't have the disease, that have the disease, but on the young people you cannot tell, you know, how many of those are going to develop it later. So you want to do this at older people. You're not going to do it in people of 40, 50. You will probably go to 70-year-old people. Okay. So what you do basically, you can imagine that for each gene, you can have a different state. You can have a 0 if it's like, let's call it the wild type or the normal state of the gene. And you can call it 1 if you have a specific mutation, and 2 if you have another mutation, okay? Then you can imagine that you can have and you can imagine you have 1 million of these, you know, and you have 1,000 individuals, okay? 1 million of these 0, 1, 2, and 1,000 individuals. And what you're going to try to see is try to find specific mutations which are associated with the disease, okay? This is mathematically very complex, as you can imagine, and statistically also very complicated. But I want just to give you the general idea. This gives you a general idea. It looks at this gene in particular. Look, it's 0 in all the controls and 2 in all the cases. So this probably is a candidate gene. Of course, it's never so clean, okay? Of course, maybe you will have, because you have you need many mutations in many genes. So maybe you have, if you have 1,000 control and 1,000 cases, maybe you will have a same state in half of them. But then, you know, they will need to share some other mutations, okay? So you understand more or less how this works? Yes? Okay. Well, this is, doesn't really matter the exact number. Usually you need more than 1,000 people. And following up your question, you need to match them for race, ethnicity, and sex. And you need to match them also for age in this case, okay? Then you're going to measure the state of these 1 million SNPs. And then you need to do a lot of statistics in order to show that this is not by chance, okay? Because you will have markers which will be the same just by chance. So you need to evaluate this probability and do statistics in order to get this, okay? This is old. I think this is from a few years ago. Now, in the last few years, there have been massive amount of these studies with there's many new technologies now to do this. Okay. 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 So there is one. Wait, okay. So the best target is apolipoprotein E. That I think that Mona also probably mentioned. This is a very protective target for Alzheimer's disease. I mean, depending on the mutation you have in this protein, you're going to have much more chance to have Alzheimer's or much less chance. So this factor is also a very good candidate for therapy because mutations there can be protective or anti-protective. And the idea that the apolipoprotein E has a lot of, could contribute to many things from the aggregation to the repair or the toxicity in the synapses, toxicity in the neurons, to formation of this extracellular or intracellular aggregate. So apolipoprotein E seems to have a big role, and you will never be able to a priori say that this gene is such a good candidate or is so involved in the disease without those studies. So you can see that apolipoprotein A could be in a few places here. So depending on the mutation that you will have, you are going to be more or less prone to have cerebrovascular events like a stroke. And we know that people who have a stroke, they have much higher chance of have Alzheimer's. So if mutations in the ApoE that are going to induce a stroke are going to go unhappily towards AD, but mutations the other way around will go against. ApoE also can be important for the clearance from the brain. So mutations that enhance the clearance function or the efficiency of the clearance of this A beta are going to go in your favor, if the other way around against. And also ApoE can be involved in the oligomerization of the A beta, which, as I said before, is a toxic form, and is the form which is more damaging for synapses in particular. So this is also important. And this was by far the best candidate. ApoE was by far the best candidate. But now you can look for the hits, with other hits which are maybe not in all the people, or not in a big proportion of the people, but that combinations of mutations that give you the disease. And then you can generate your ranking. You know, the most common ones, the less common ones. And the most common ones probably are the ones which are more, like when mutated, can give you more chances to get the disease. And this is just the list here. Okay. My idea is to try to go fast, one by one over them, and try to tell you where they fit in this picture. Because this model I built at the beginning is only, we didn't know this 20 years ago. This model I built in the beginning is based on all these hits of all these studies. Okay? So once we get the data of the candidates, we can make a model and try to understand how the disease is produced. So that was the question. Okay. So the first one is this protein called ACE, or angiotensin converting enzyme. And this is very important for blood pressure. And this, we know that mutations in this enzyme increase 20% the chance of the disease. And again, this suggests blood pressure has a role in the disease. So then, based on that, people can do intervention and try to manipulate in low. We know that having high blood pressure is bad for you, okay? But also this can be important in determining Alzheimer's. Okay. So basically, in the case of ACE, ACE is very important for the clearance of the aggregate. It's here, sorry. And they also, because regulate blood pressure, has been trying to be used as a therapy, not extremely successfully. Okay. So you also can, indeed, a mutation in ACE also can increase, of course, stroke, because it's related to blood pressure. So the second candidate was this cholesterol enzyme. And in this case, cholesterol may be important here for how APP is processed. Okay, APP is a membrane protein, so cholesterol is important for the kinetics of this. And also the clearance of the aggregates can be regulated by the cholesterol. And also, of course, cholesterol is important for the cerebrovascular events. Okay. So then you have this nicotinic acetylcholine receptor. In this case, if you now mutate the same enzyme, you have what we call Alzheimer-like mice, which have problems in memory and recalling events. So it seems like a good target. Okay. So and what we believe is that in this case, this enzyme is involved in the final stages of the disease. Okay, and also in the tangle formation. Okay. Okay. Then you have other protein, which is it's an enzyme that is an inhibitor of cysteine proteases. and which must be involved in also in some of the stages of the disease. I don't want to go to the detail, but what I want to tell you, what is the pathway people do? You do a genome-wide study, and this tell you and teach you how the disease happens. Okay. And then give us possibility to intervene. And happily not very successful yet. Okay. Okay. I want to go. This mamash not really important. Okay. We're going to go to the whole thing. Okay. Okay. So basically now we have a lot of genes which we believe are important for the disease. Okay? This allows us to make a model, okay, of the factors which are how the disease starts, how the disease progresses, and how the disease finishes or kill you. So this now, of course, if we want to try to design therapies or to design approaches, we cannot do this in a person, because you cannot just you say looks to me that the ACE is important for degradation of the peptide. This is related to blood pressure. So maybe we just manipulate blood pressure. Let's take 20 guys with Alzheimer's. Let's give you some drugs to manipulate blood pressure. You cannot do that. So, but you can do is now to build models, animal models, that will allow you to address these issues. Okay. And that's where I'm going to go today. Most people use animal models which are mice. This is a very common model for many of these diseases. Now, the mice has a problem. Imagine now we have a lot of these studies. Imagine that you have 200 or 300 candidates. Okay? So how you cannot do 200 or 300 mice in a reasonable time. This takes a lot of time. So how people have been using in the last few years other type of animal models which don't don't need so much time, and they have been using models. One of the models is a fish called zebrafish. This is a vertebrate, as all fish, so it's not so different than us. And the other type of model is flies, which also are not so different than us, though we believe we are much better. And other models is worms. So all these models have been extremely successful, and in some cases have helped us to screen towards from hundreds or thousands of putative genes and find new pathways in neurodegeneration. Okay. You guys finished with the list of the people? Who has the list? Okay. Just give it. No, because we will pass it in a few minutes again. Eh? Just give me this. Okay, thank you very much. Okay, so I will also forget. Okay, so these are the animal models, the fly animal models. So the question, why? I'm going to talk today about using flies as a model for neurodegeneration. I'm going to try to convince you that this is a good model. I mean, I don't need to convince other scientists or funding agencies, because this is a very popular model, also used by Novartis and other companies to screen for compounds and things that could not be done in mice. Number one, they have a very short half-life. If you have your favorite gene that you want to make in mice mutated for, it will take you a year to have these mice. From today, it will take you a year. If you want to do it with a fly, it will take you maybe 3 weeks. So if you want to screen 100, in a few months you can screen your 100 candidates. If you want to do it in mice, not only we are going to spend a lot of money, ridiculous amount of money, but also will take you years to check. In this sense, maybe a better strategy is to check in flies in a few months and then make the mice which seem more promising. And that's something many people are doing.
But how can you tell if flies suffering from dementia or memory problems?
Yeah, we're going to see, I mean, the pathways which are regulating the neuronal maintenance or survival are very similar. Very similar. What happened in Alzheimer's and Parkinson is that the specific neurons die, and the neurons that die is the one taking care of the memory. It's not something about the memory itself. It's about the type of neurons who die. So if you can identify the factor that would kill the same type of neurons in flies, this is going to be very similar. So the first thing is that they have very short life cycle, so this is some sort of advantage. The second is that despite we believe we are so complicated and so the most sophisticated thing in evolution, which maybe we are, but the neurophysiology of the fly brain is very similar to our brain. And indeed, if there is some databases in which you can look for the genes involved in disease, most human genes which are involved in the disease, they also exist in flies. Okay? Indeed, people have generated so many models. Many of those models, if you mutate the protein which is the same in fly, you're going to get the same phenotype. This is true, especially for cancer models. In flies, you have, now in mice also you have, but in flies you typically have great genetic tools. Basically, usually with little effort. I don't want to go into detail, but basically, if you have a candidate gene, you can overexpress it and knock it down very, very easy. And you can overexpress it and knock it down in very few cells, the one you want to, and when you want it. So you have very easy ways to manipulate gene expression in specific patterns. Okay?
Can I ask?
Yes.
It's a little bit off topic, but it's very interesting for me to know. Do you talk about the moral aspect of experimenting in animals? Because I know in my area, which is humanities, we always speak about it. And I wonder if you scientists ever raise up the question.
That's a very good question. More interesting maybe than this. I should say that there is people's draw. I mean, this ethical line always can be kicked further and further and further. I think this type of debate should be, especially for people working on stem cells, especially human stem cells. But we always believe that in the case of the research, because our will is to increase. I mean, it's a selfish interest. Okay, of course, it's selfish interest of humanity to or to people to live longer or live healthier. But I would say that our ethical line is that whenever we believe our research is going to improve the quality of life, it's worth it, unless it's giving suffering to the animal. I don't know if our assumption is that our research is to help the human race, let's say, to call it somehow, and then that is justified to do it, unless the suffering is to the animal is too much.
How do you know if they suffer?
Yeah. I mean, of course, in animals which you can relate more, like a monkey, you can understand it better. I personally never work with monkeys, and I will not do it. But maybe I could help much more people by working with monkeys. Maybe it's a selfish decision to do it. In particular, I also don't work with mice. I mainly work with flies or with cells. But I should say that I have an experience when I was doing my postdoc that Indian colleague didn't want to kill the flies because of this. Indeed, I respect so much his decision, because he told me, the flies have a karma, and you could be reincarnated as a fly later. But and I saw this is system of believing maybe as valid as mine in science. So I respect, but I don't have the same, so I could kill them for him, and then he will not have this problem. But this is, of course, we think about this all the time. We do this assumption, and it's true. We don't know they suffer or not. And I think that in some diseases like Parkinson, the advantage of working, the question is, sorry to go off topic, okay? I just found this interesting. So I will 2 more minutes and we go there. It's not so off topic though. So in Parkinson, it's clear that working with monkeys help a lot the research in humans. So the question is here, well, then what to do? I mean, I don't know what you guys will do. Because work on monkeys in the electrical stimulation has been very important to use this as a therapy in humans. And this is the only therapy that works for Parkinson and works quite well for the movement disorder indeed. You know, deep electrical stimulation in the brain works very well in Parkinson. Is now applied much widely. And probably a combination of this with something else may eventually cure it in the future. And all this research happened only because working monkeys. So I think that it's a decision which is quite hard that I would not be able to take at all. So, but again, it's opposed. You know, one view see the human being as more important than anything else, and more moral. I mean, I'm not sure that the morality is something that we choose or something that comes with us, and maybe our instinct will be to survive. So our instinct will be to do those experiments. So I'm not sure whether we need to overcome it or not. Now, I should say, talking in this issue, I will of morality. Now, huge debate will come because now we can really, we can probably clone humans now. It's something that the technology is there. And the question is whether to, I wonder what will happen here, because we can take a skin cell from a person and make a stem cell, and from this you hypothetically could do it. If it's not now, technically it will be a question of years. So I think that a lot of moral things will come there. On the other hand, you can imagine that the fact to clone a human can give you power to do transplants or to do things which potentially could be very good, you know, and also to cure diseases, because if you don't know what makes a Parkinson person to get Parkinson, then you can take a cell from this person and study this in more detail. So, but it's good, yeah.
I want to ask, in order to get a cure, for example, okay, we found a problem, we made a medicine or something like that, we have to do the checkups on all the living species there is. Like we need to
you mean if you got it in fly?
Check it on mice,
yeah.
And we need to check it on monkeys, or
yes.
check it on flies and then.
I mean, you have a procedure that the procedure for releasing the drug involves trials on animal model, and then you have a lot of toxicity trials, and then you usually need to do it in a primate, and then in a human. Yeah, you usually need to do it, but the path is much shorter when you pass through one of these animals at the beginning, either C. elegans or flies. I mean, and you need to understand that there are so many drugs already approved by FDA that what people is doing now is trying to see with any of the already approved drugs, which we know they are not toxic, they work in any of these animal. It's a complete blind bet, you know. But you can now go much faster because if the drug is approved, all the toxicity assays, you don't have to do them, and you can basically maybe show it in fly, show it in the mouse model, and that's it, you know, because it's already approved for something else, you know. Many of the drugs approved for diabetes seems to be also working in Parkinson. It is not clear completely why, no, but yeah.
If they mix like two different stuff, for example, there is something for diabetes and something extra that we never checked. I don't know, like alpha amyloid.
Yeah, no, you need to show the two together is better, and you need to do all that. Yeah, you have. I mean, again, this is you know, you don't have what you do, you know. Yeah, I mean, there is so many arguments. You know, from the ethical point of view, it seems to be like impossible to solve. You know, it's and again, people talk about this line that you keep crossing, you keep moving the line. You know. And that's a question whether you should move the line or keep the line and just not do it, you know? Especially with the stem cells, I think it's a big question. Okay. Any other question? Okay. So how we model neurodegeneration in Drosophila? One way, when you have recessive ways of a disease, you can basically, if you know that the protein A causes Parkinson in humans or in mice, then you can just take protein A and just mutate it, let's say, downregulate it, okay? This is when you have a disease which is recessive, when you need your parents to have it, okay? Okay, of course, in flies, you can only model a sporadic form of the disease, which are unigenic, one gene. If you have a dominant form, like Huntington, then you just express the dominant protein, the human protein that causes the disease, and this usually recapitulates well what happened. Okay, this is really not really important, okay? So where you express this? So the good advantage of Drosophila is that, you know, if you imagine that you need to express this in the brain and follow the learning of the fly can learn things, but it's very difficult to teach them. So it's going to be very difficult to follow this model. So but what people have done is to express this toxin only in the eyes. Then when the fly really have death of the neurons in the eyes, you can see this. Instead of seeing the normal eyes, when you express a toxic protein, which is a little toxic, you see now it doesn't look so nice. And we express a very toxic protein, now the eyes will look like crap, okay? Will look really bad. And this gives you a very powerful situation, because you imagine now you have a fly which the eye doesn't look good, okay? Because in these eyes you model Parkinson's disease. These neurons are dying there in a similar way that the dopaminergic neurons in our brain are dying. So now you have this fly, which you can very easily see how quick and how you don't have to kill it. You can follow it. You can see day 1, day 2, day 3. Now you can feed this fly. You can take 10,000 of these flies, and each one you feed with another drug. And then you can find drugs that make it worse or make it better. You can find drugs that maybe delay the disease, not just that don't provoke it, but delay it in time. So this is a very powerful thing, that you can use the flies as a test tube for the most basic aspect of the neurodegeneration, which is just the cell death. Okay? Of course, you cannot follow memory here. You cannot do sophisticated assays. But this will tell you briefly whether the neurons which are dying by the same cause of Parkinson can we delay it or make it faster, or et cetera, et cetera. So that's what people have been doing, and I don't want to go into detail because you know, you guys will get lost in the details, and the important thing is the general picture, that if you have a fly model of the disease, you can now try a lot of drugs to see whether they have an effect. And you can also mutate every single gene to see whether the combination of the first mutation and other mutation make the disease to be better or to be worse. Okay? Any question? No? Okay. I don't want to go into detail, but here is one. This is a poly-Q disease, and this is the control. This is also a fly. Now, when you have a sick fly, you can see the structure of the eyes. It's not as compact. Here you have these holes in the middle, and as the time passes, you see how these holes extend. Okay? But you can also see it from outside. This is the structure of the eyes. Okay. I don't want to go into these details. I'm going to skip this, okay? Okay, so basically, I hope I convinced you that these type of models are better. And if you think in using animal models, I mean, using flies will be, you will use much less mice, let's say, in one hand, okay? Okay. So you learn a little about Huntington, no? Who teach you Huntington? No, you didn't. Eh? You didn't. Okay, so I will, in the last 15 minutes, I will tell you a little about it, okay? So many neurodegenerative diseases, I saw that you heard about it, not at all. But I know you didn't have a class on it, but you heard it about it?
No, we had a class.
Very general.
Very general, okay. So many of them, so then you can make me go fast, and I go to the more deep stuff if you didn't see it. Okay, so many diseases show this anticipation, and this is that, you know, if you have the disease when you are 60, then your kids have it when they are 40, and then the kids have it when they are 30, etc., etc., okay? And then the disease will disappear one generation and will appear again, okay? And one of these is this type of genes, which involve repeats of 3 nucleotides, okay? And these are specific 3 nucleotides in the DNA sequence, which are going to lead to specific repeats in the proteins. It doesn't matter, but these repeats are these repeats CTG specifically, okay? There are many type of disease involving these 3 nucleotide repeats. But this disease can be distributed into 2 groups. One, when the repeats appear in the protein. And this is the example of Huntington, okay? When you have a repeat of a sequence in the protein. And other type of cases is going to be when the repeat happens in a region of the gene that is not in the protein, okay? And this is like Friedreich ataxia. We'll give you an example of it. So how and this coming back to the same point, how do the expanded repeat cause disease? So I told you that some of them, many of them, HD is Huntington. Many of them happen in exons. In exons are the part of the protein which make a protein, the part of the gene that makes a protein. And this you can tell me, okay, what happened now is this repeat makes a different protein which is toxic. This makes sense, okay? Yes or no? Yeah? When you have you have this expansion in the coding sequence of a gene, this makes a protein that is toxic, okay? Now, you can also have the repeats in regions which are the promoter of the gene. This is the region that determines how much you make of a gene. So this type of mutation is going to act usually as a deletion of the gene. Because if you have this expansion here, in this region, the promoter, then the gene is not going to be expressed anymore, okay? Why this face? So you know that we have a copy of the gene from the mother and from the father. In the case that you have this expansion in the exon, one will be enough to get sick because you are making a protein which is very toxic. Okay? So when you have the mutation in the exon or the expansion in the exon, then you are going to have the disease. Even if you have only one copy of the disease, these are dominant disease like Huntington. Yes?
What happens to the person? Which sick in it?
Okay.
How it inflects on the person?
Yeah, I mean, usually what you have is ataxia. This is a movement disorder. Okay. You have degeneration of the brain, starting from.
Parkinson also.
Yeah, Parkinson also, but in Parkinson you have a 3 nucleotide repeat. Okay. We're talking now about this type of disease in which you have a 3 nucleotide repeat. Okay, and leads to phenotype which are similar to Parkinson. Yes, you have first a movement disorder. You can't move. Then in many cases you have also mental retardation or a mental impairment, dementia. Okay, depends on the thing. Different depending on the disease, different type of neurons die. So some of the symptoms are a little different, especially at the beginning. But in most of them, at the end, you die earlier. Okay. Speech problems also, you know, depends exactly of the disease. But this is the idea. This is a general symptomatic. But now this expansion basically is that now these genes are going to have a huge piece that was not there before. If this huge piece is in the exon, is in the coding sequence, then you have a protein which has a huge region which is now toxic. So doesn't matter if you only to have from the mother or from the father make you to have the disease. This case is a dominant disease. Now, also you can have this expansion in what we call the promoter. The promoter is the region of the genome which is just before the gene. And this region is basically important to determine how much of the gene you're going to make. In this case, when you have these expansions there, now you don't produce the gene anymore. It's like a deletion. It's like this is gone. But now, if you delete it from the father, you still have it from the mother. So in these cases, when you have the expansion in the promoter, you usually have, it's a recessive disease. You need to have both of your parents sick to have it. All these are Mendelian disease, okay? They are not sporadic. You can also have it in this sequence called introns, or in the 5' here. It doesn't really matter. It's also non-coding, okay? So you can have it in coding sequences. When you have it in coding sequences, you have proteins which are toxic. When you have it in non-coding sequences, usually it's like having a deletion of the gene, and usually it's recessive. Okay. Any question? No? So this is a very these are many, many different types of diseases. And if you go to a book maybe 50, 60 years ago, they will not be related at all because the symptomatic is different. I mean, not exactly the same. The disease looks different. They run in family, but not in the same family, in different families. So they don't seem to be related. Only now, in the last 40, 50 years, that we could associate what happened. We realize that all of them come from expansions of a specific sequence in the DNA. Okay? And some of them are untranslated. And these are the ones who happen in promoters or in regulatory regions. They don't happen. They don't lead to the production of more protein. Okay. There's one of them. The most common one is one called Frederick ataxia, which is highly uncommon, and it's a recessive. Okay, it's 2 to 4 cases in 100,000 people. Happen unhappily when people are very young, less than 40 years old. The people gradually lose movement capability. They need to be in a wheelchair, and they die around 50. Most of them need to be homozygotes to for this expansion to have the disease. This means both your parents need to be carriers. It's a recessive disorder. I don't want to enter into the details of how this happened, but I think it's interesting. This disease, basically, you have you lose a protein called frataxin. You lose a protein, which is very important to maintain the iron homeostasis in the cell. Okay, the iron, the cytoplasm, and this protein seems to be transporting it. So what happens is people they accumulate a lot of iron in the mitochondria that cannot get out anymore. So a lot of enzymes which needed the iron in the cytoplasm, now they don't have it. And then they have oxidative stress, mitochondrial loss, and eventually cell death. Okay. Then you have other type of disease in which you have a gain of function. Again, all these type of disease, you have these nucleotides that expand. So they have a huge new piece of DNA, which is, it doesn't matter how it's generated, but you have in these people a huge piece of DNA, which now is provoking the disease. When this is in the coding region, you got a protein which is completely abnormal. And this is the case we're going to see now. And yeah, this is the same as before. Sorry, yeah. I don't know why. I upload the wrong. I delete all these things, you know. But I think I uploaded the wrong presentation. Okay. I mean, it has some extra slides that I think are too technical for this class. So you have many other diseases in which I said these 3 nucleotide repeats happen in the coding sequence. So now you get a new protein which is very, very toxic. Okay? And this is the example of Huntington disease. Usually people have this repeat between 6 and 30 repeats. But in the people with the disease, you can go up to 120 of these repeats. So these are just some of the clinical features. Okay? But the idea here is that you have a sequence which is in the DNA that now gets expanded 2-3 times in length. And this provokes the disease. Okay, so these are autosomal diseases. They are dominant. This means you need only one of your parents to have it for you to have it. They have this expansion. They are also called polyglutamine because the sequence CHG in the DNA is translated in the amino acid, which is called glutamine. So you have polyglutamine now that were not in the protein before. And you want to go to one of the most important things is how these diseases are produced at the molecular level. So when you have a protein with this poly-Q or polyglutamine, Q is the name of glutamine when you do amino acids in one letter. So polyglutamine disease or poly-Q disease, you have again, and I want you to remind you what we talk when we talk about Alzheimer's or Parkinson. We talk about this aggregate, these balls of proteins, which now are toxic, probably toxic for the cell. What happens is that this protein is fragmented, and now this glutamine, they like each other. They have affinity to each other. They have chemical affinity for each other. So they're going to aggregate in these huge aggregates, first in this fragment, which is going to lose the conformation, and now this glutamine will tend to interact with each other. Now these oligomers or microaggregates are extremely toxic for neurons, especially for synapses. The cell is going to try to get rid of them either by degrading them, as I talked in the first class, or by taking them out of the solution, forming this big inclusion. So what is toxic here is not the big inclusion that you see in the microscope, but the oligomers that you don't see them really. But of course, the cell has a limit, how much can take of the solution. And if there is a big production of oligomers, then it's going to win and kill the neuron. And I think that if you, again, like in the case of Alzheimer's, you see this as a quantitative process and not a qualitative process, you will understand why these diseases don't happen when you are a kid or when you are 2 years old. Because when you are a kid, you still can deal with these oligomers and get it out of the solution, or misfolded back and degrading. But little by little, these things start to accumulate. Your degrading mechanism start to deteriorate. And when you are 30, 40, you start to actively accumulate in these oligomers. And you cannot take them more of the solution, and then it's sort of irreversible. But again, will depend with age, your capacity to fix this issue. And this probably related to the age dependent, or the fact that with age, you get more propense to the disease. Okay. Okay. So one aspect that I want to mention, I told you that when you have, I told you if you have a poly-Q disease, then your kids likely will develop the poly-Q disease earlier, and your grandkids even earlier. And again, this has to do with the length of the poly-Q. So when you have a length of the poly-Q of 40 or 50, now your kids are going to have a longer poly-Q. And this has to do with what happened with the sperm or the eggs when it's reproducing. When it's dividing, it's going to produce this longer. It's going to commit mistakes. When you have this poly-Q, once you have expanded poly-Q, each time you are going to duplicate the cell, it's going to commit more mistakes and make it longer and longer. And that's why it's progressive. The kids have longer tracts than the parents, etc., and this increases the chances that you will have the disease earlier. Okay, that's why this anticipation happens. That as you go in the generations, you get the disease younger. Okay, so I hope that I could deliver. My main message today was to try to tell you how we do research in neurodegeneration. We try basically to first go to the human and try to see which genes are affected in the human. This helps us to build a model, hypothetical model, how the disease progresses. Help us to try to rationally design therapeutic approaches. Help us to rationally use animal models to test these therapeutic approaches first. Using these animal models, we can also look for other drugs or genes which are important for the disease. And from the conceptual point of view, this type of approach has taught us that the neurodegenerative diseases are something which is quantitative rather than qualitative. maybe all of us supposed to get it, but depends how those pathways are affected at the quantitative level when we are going to get it. If it's in our lifetime or it's after we are dead. So the challenge now comes to use all this information in order to change our habits or to find medicines to delay this thing. Okay? Any question? No? Okay, guys, thank you.